Welcome back to my channel Xgene proteinX. In this tutorial video, I'm going to talk about the CRISPR-Cas9 system. So first, you have to know that what is CRISPR. So CRISPR, in short, it is known as the CRISPR. So the full name is actually the clustered, regularly interspaced short palindromic repeats that found in the bacteria and archaea for prevention of the infection induced by the phage virus. So this is the CRISPR. So if you have a closer look this figure, then I think it might be more clear. So that is a clustered, regularly interspaced short palindromic repeats. In short, that is the CRISPR. So this is the CRISPR assemble. So CRISPR assemble contains spacer sequence and some interspaced short palindromic repeat sequence. That is this kind of repeat 1, 1, 2, 3, 4. There are 4 repeat sequence. So the spacer sequence is nothing else. This is the copied sequence, copied short sequence from the foreign DNA. So I'll discuss later what is the spacer sequence. So this is the CRISPR assemble. So now you might have a question that what is the palindromic repeats? So let's have a look at what is palindromic sequence. So palindromic sequence is the sequence of letters read the same left to right. So for example, so if you read this sentence never odd or even. So if you read this sentence from right or from left, so it will be the same, right? So this is the palindromic. So it's the same things in the sequence as well. So if you this sequence actually GGATCC, and in the complementary it is a CCTAGG. So if you put it beside, then it might look the same, right? So if you read this sequence from the left or from the right, it will be the same. So this is known as the palindromic sequence. So let's move back to the where we were. So we are here. So that is why this kind of sequence are palindromic sequence. So this kind of sequence contains some repeat sequence and some this sequence actually 23 to 47 base pair long repeat sequence. So here it is 1, 2, 3, 4 repeat sequence, and in between there are some spacer sequence. So now you already know that what is CRISPR. So you have to know right now that what is Cas9. So Cas9 is the nuclease enzyme, and that generate double strand breaks at specific region of the genome. So this Cas9 it recognize the DNA RNA duplex or DNA RNA hybrid. That DNA is the foreign DNA, foreign virus DNA, and it detect this hybrid, and then it cut or it cleave the foreign DNA in certain specific sequence. So this is the job of this Cas9, and this is the Cas9. Alright. So now let's have a look at what the history of this the discovery of CRISPR-Cas9 system. So first is very beginning, the Francisco Mojica. So one of the scientists, the Spanish scientist, and he found the CRISPR sequence. So he is known for this repetitive functional DNA sequence in bacteria that is known as CRISPR. And in 1993 he identified that this kind of repetitive sequence in the genome of the Halopharax, one of the archaeal. He found this kind of repetitive sequence. And later on he also observed this kind of similar sequence in prokaryotes, as well as this sequence it matched with the genetic material in phage virus. So this kind of things he already observed a couple of years back. And later on in 2005, Mojica hypothesized that these sequences are part of the microbial immune system, right? And then in 2012 the duo published their experiment showing that the CRISPR-Cas9 system could cleave isolated DNA. So this is all about the previous scientist discovery, what they found. And very recently, 2020, Dr. Jennifer, he was the first scientist to propose that microbial immunity mechanism could be harnessed for programmable genome editing. So she was the first scientist who observed it and who did and who proposed or proved that this kind of genome editing, this kind of CRISPR-Cas9 can be used for programmable genome editing. And also another scientist, Emmanuel, she also co-inventor of the Cas9 enzyme. And both of these scientists, they awarded jointly Nobel Prize in 2020. So let's have a look at what is the mechanism of this CRISPR-Cas9 system. So first, in the CRISPR-Cas9 system, there are two different phases: the acquisition phases and interference. So in the acquisition phases, where the bacteriophage, it attack any bacteria and then incorporate its genome into the bacteria. So next is the after infection of the DNA sequence from the bacteriophage and then into the host CRISPR locus of the spreads of the spacers. So after incorporating this genome from the virus, then some of the small sequence of this foreign DNA, that is called spacers, and that assemble into the CRISPR locus inside the bacteria. So you might have a question that what is CRISPR locus? So CRISPR locus, actually this is the assemble of some different kind of sequence. So it contains some Cas genes, that is, I already mentioned the Cas is Cas9 or other kind of sequence, other kind of enzyme. So basically the Cas9 here, and also here is the Cas9, and in between there are some sequence, that is the spacer sequence, as well as some palindromic repeat sequence that I mentioned in my previous slide. So this is the CRISPR actually, and this sequence is actually the CRISPR, and these are the Cas gene. So this is actually the CRISPR locus. So whenever this foreign DNA is incorporated, then some part of the sequence it chopped and then it assembled in the CRISPR locus, and then as a result the CRISPR locus is transcribed and the precursor CRISPR RNA is formed. Alright, so this is the precursor CRISPR RNA, and then the precursor CRISPR RNA is cleaved to produce individual CRISPR RNA that will associate with Cas9 proteins. So as you see here that this small fragment of the CRISPR RNA, it cleaved individually from the precursor CRISPR RNA, and then it associated with Cas9 system. And then the next phase is the interference phase. And interference phase, when the same virus, when it attack the bacteria the second time, then the CRISPR-Cas9 complex is able to identify and silence the foreign DNA. So as you see here, this the CRISPR-Cas9, and it contains the RNA. So this is the RNA, and this RNA, it matches with the foreign DNA, and then it bind with that complementary sequence of the DNA, and then the Cas9 involved with this system, and Cas9 detect and recognize this DNA RNA hybrid, and specifically it cleave the foreign DNA. So this is how it works. So the sequence, the short sequence, that is the short DNA sequence that is targeted for the cleavage, that sequence is called the PAM sequence. That is the protospacer adjacent motif. So let's have a look what is the PAM sequence elaborately. So PAM sequence is here, as you see in this picture, that this is a target DNA, this is a foreign DNA, right? So here it contains this kind of small 20 nucleotide sequence that is matched with the RNA, and that RNA is the guide RNA or CRISPR RNA. And when this CRISPR RNA and the DNA duplex formed, then Cas9 it comes here and it recognizes this RNA DNA duplex, and then it cut some DNA sequence that is 2 to 6 base pair long, and this DNA region is targeted for cleavage by the CRISPR system. So this short sequence that is follows the DNA region targeted for the cleavage by CRISPR system, that is known as the PAM site. So this is here, this is the red red sequence of this sequence is known as the PAM site here. Alright, so this is all about the CRISPR-Cas9. So in my next video, I'll discuss about the gene editing system by using CRISPR-Cas9. So if you find this video helpful, so kindly hit the like button, share it, and subscribe my channel. Thanks.